So thanks very much, David. It's a delight to be here and see so many new faces. This is a cool society, I think. And when we had the opportunity through the Stem Cell Institute to serve as sponsors, we were glad to do so because excuse me, this is the kind of thing that we want to foster, where the ideas of fundamental science are put into motion and, in fact, end up making their way into the clinic in various ways. And so I'll tell you today about my aspirations to regenerate the heart, and this has been like a 20-year saga. I'll spare you much of the pain and suffering that we've run into along the way. I would also like to encourage you while you're here, get out, stretch your legs a little bit. This is a gorgeous part of the world, and for those who won't see it too often, the weather should be beautiful, and so there should be lots of fun things to do. We're known for coffee. Starbucks is of course based here in Seattle, and so there's many of us who are fairly addicted on the coffee front. Washington is the second biggest wine producing state in the nation, second behind California, and there are great Washington whites and reds that are available. We're replete with craft breweries. If people are inclined like I am towards India Pale Ales and things like that, there's all kinds of ways that you can indulge those vices and that sort of thing. And I won't. And we passed a law in 2012 legalizing some other things, but it's not fit that I speak of it here from the podium. But it's an interesting place, and so I just would add my welcome to that of David's, and say enjoy your time while you're here. I have a disclosure. I'm a scientific founder and an equity holder in a company called Cytocardia. That's a spin-out company that is about to bubble out of the University of Washington. And its goal is to take into the healthcare sector some of the work that I'll talk about today. And my other disclosure is we don't have any money yet. And so that's the sad part about my disclosure. But it is always, of course, good to A, disclose, and B, to keep it in mind. The most important thing I'll probably say is here, I don't want to run out of time and not be able to say thanks to the people from my shop who made this possible. This is an extraordinary group of people whose work I get to represent. And so along the way in my presentation, if I say I mean we. If I say we, I mean they. I mostly sit in the office and talk on the phone. I mean, it's like, what do I really do? I don't know. Things run down if I'm gone too long. But anyway, these are the heroic folks who get all the work done. Okay, with that, let's just launch into it. I think you all know that heart disease is really bad and that you don't want to get it. It's now the #1 cause of death across the whole world. This is as of about 15 years or so. The global burden of disease has shifted from, as you all know, from infectious diseases to chronic diseases and so forth. And here in the States, it's the #1 reason people are admitted to the hospital. It's #1 Medicare expense, the #1 private payer expense, that sort of thing. So this is the big thing that is really, and it's burgeoning. It's a burgeoning impact on public health. Hundreds of billions of dollars clearly are spent on direct care and even more on indirect loss of productivity, caregiver expenses, that kind of stuff. Here, I'm a pathologist, by the way, and so I have sort of a pathological perspective on things. This is the pathophysiology of ischemic heart disease. Heart with a coronary artery that you see here the artery becomes obstructed by an atherosclerotic plaque. Typically, an acute event happens, like the plaque breaks down and a blood clot or a thrombus plugs off the artery. The muscle that is fed by this blocked artery then becomes acutely ischemic. And in the heart, it's one of the most metabolically active organs in the body. And so you get about 15 minutes before you start to have irreversible injury, cell death. And then this wavefront marches through the wall of the heart so that you lose all the heart muscle you're going to lose by 3 hours. About 80% has died off. By 6 hours, about 100% of the tissue that's at risk will have died. So this is a very fast event. We have great tools to move people in once they come into the emergency department doors, we get them into catheterization laboratories, and we open their arteries with clot-busting drugs and balloon catheters. And this has all been Toyotified. We've got, the door-to-balloon time is down to the limits of human endeavor, I think. So that's terrific. The rub is, of course, that most people wait way more than 3 hours before they seek medical attention. And this has had us tearing our hair for 20 years. And until one of y'all isolates the denial gene, this may continue to be the case. So the heart, unfortunately, is one of the least regenerative organs in the body, probably vies with the central nervous system for who is least able to regenerate after injury. The problem then is the heart heals instead by growing back scar tissue. So this dark area here that's depicting a myocardial infarction will be replaced, it will be forever lost as muscle and will be replaced by scar tissue. The scar, of course, it does not generate contractile force. This leaves the heart with a contractile deficit. And you don't get just a stable, new, steady state. Unfortunately, you get an unstable state that has a rundown associated with it. And so the patients gradually spiral into a decompensatory process that we know as heart failure. Once you're diagnosed with heart failure, it's quite deadly. You have, on average, about 5 years to live. So really deadlier than most forms of cancer. So it's a classic disease of cellular deficiency. So our notion is pretty straightforward. Let's repopulate the wall of the injured heart with stem cell derivatives and see if we can remuscularize the wall. I had this idea 20 plus years ago, and I thought 5 years we should be in the clinic. And we're finally now getting to the point where it's actually feasible. And so first, so much for my judgment, right? My ability to predict the future. I thought everything was going to be easy. It hasn't been, but we've been making steady progress. Things picked up. We started out, we didn't know how to differentiate stem cells into cardiac muscle. We learned how to do that, and the cells we'll be working with are pluripotent stem cells, the most potent amongst the cell populations, either embryonic stem cells or induced pluripotent cells. They're immortal. They can be grown up in essentially inexhaustible numbers, and we know how to differentiate them into cardiac muscle cells, or cardiomyocytes as they're known. We went up through the food chain. We learned how to transplant them into small animals first, mice and rats, and we got that working. We moved up to guinea pigs. This was taking really long, as I said. So a number of years ago, we made the decision to leap into large animals, and we picked the non-human primate, the macaque monkey, as our subject, in part driven because we have this fantastic primate center here that was already very good at transplantation biology and immunology, HIV/AIDS, that sort of stuff. And so we had a lot of infrastructure upon which to rely. This was work that was led by a talented postdoc named James Chong. And the major findings from this study we published back in 2014 was that we could get good grafts of human cardiac muscle. Of course, we used 750 million cells that we delivered, and so you should get good, mass in, mass out, essentially. The cardiac muscle that we transplanted coupled electrically with the heart, and I'll show you that in just a minute. It matured to being pretty good-looking adult-like muscle. I'll show you some of that. It became vascularized, probably not as good as native tissue, but decent. And here was one of the rubs. It made the heart twitchy for a while after we transplanted it in. The heart, there's a couple of weeks of electrical instability, ventricular tachycardia, that sort of thing. We hadn't seen that in smaller animals because their hearts were too fast. When we got into slower species that were more akin to humans' physiology, then we found it. So we were depressed for a while. Everyone was on serotonin reuptake inhibitors. But then we're like, well, I mean, good catch. We didn't do this in someone's mother. We caught it. It was just a bummer. So the questions that I'll be drilling down on are twofold. The first is, it's cool that things look good under the microscope, but does it actually benefit the physiological function of the heart in this human-like animal, number one? And then number two, what the heck's going on with these arrhythmias? Why is the heart electrically unstable after we transplant our cells in? So we did this, what we call this efficacy study. This was just published over the summer, and it was led by a triumvirate of postdocs, each of whom had about a 2-year stint at the helm. This is a really long, very expensive study. Yenwen Liu from Taiwan, Billy Chen from the University of Washington, and then Zhulan Yang, who's a permanent staff member in my lab. We induced the myocardial infarctions in these animals using a balloon catheter. Typically, you use catheters to open arteries, but perversely, you can use catheters to close arteries also, and so that's what we did. And we gave them big infarcts with a 3-hour occlusion in the mid-level of the artery. We learned how to do magnetic resonance imaging scans so that we could get the best possible structure and function of the heart. And we put in a whole lot of cells. We're trying to remuscularize a wall. We think this is a problem where you need mass. And so we needed something on the order of what had been lost. And so we picked a dose of 750 million and then Then we prayed that that was a good, a good choice. I won't get into the details of how they're manipulated, except to say that the Wnt pathway is like everything in developmental biology, and we tweaked the Wnt pathway using small molecules to control cardiomyocyte differentiation. We gave the cells surgically with an open thoracotomy, instrumented the animals for telemetry so we could track their rhythm on a 24/7 basis. This is what the infarcts look like. This is the red area is scar tissue stained with a reagent called Picrocerius red, stains collagen. This is the back wall of the heart, so that's healthy muscle. And so you can see these were big chunks. These infarcts averaged about 25% of the left ventricle. That's not what I wanted to do. I think I hit the black screen button. And this is an example of our magnetic resonance imaging. You can see a little cine if the movie will play. There it goes. And this is left atrium, left ventricle down here going out to the aorta. And apparently it's not looping. We seem to have a graphics card problem. Anyway, it doesn't matter. You got the idea. Something moved, and this is how we measured the amount of, and the thing we're going to measure is how much blood gets squeezed out of the ventricle with each beat, and that's called the ejection fraction. When we did our first study, it was brute force. We did not have sophisticated cell manufacturing capabilities. We grew everything in 2 dimensions, and we just did what was called scaling out. So this was floor to ceiling of the incubators stacked up, just liters and liters and gallons of cell culture media, and then a team of really great people just feeding. And this was ugly, but it worked. And you know, sometimes brute force. I mean, we have it for a reason, I guess. This is what things look like in 2018. We have advanced this into essentially pharmaceutical grade cell manufacturing, where we have good manufacturing practices that are in place. We go back, you know, we try to do as much as we can under like sane conditions where we're not in spacesuits and that sort of thing. But we can do really GMP level cell manufacturing, which is what we'll need to do if we're going to take a shot on goal and try this in human patients. So this is all clean room based stuff now. The other thing that I'll show you, and this is how we're now differentiating these cells. First, the pathway. We start out with an embryonic stem cell. This would work with an induced pluripotent stem cell. We activate the Wnt pathway. We make mesoderm. We repress the Wnt pathway, and we make cardiomyocytes. This is not too clear, but it's a closed bioreactor system. At a 3-liter scale, we can get about 2 billion cardiomyocytes per liter now. So we're actually more like a billion, but whatever. We grow large vats of cardiomyocytes. And I'll start this movie. And it's a little hard to see. These are little cardio balls, and we grow them as 3-dimensional spheroids. But you can start to see each of them is twitching individually. We engineered these with a jellyfish variant. It's a GFP variant that's also a calcium reporter. And so this tells you what their calcium transients are doing. Of course, every time the heart beats, it's triggered by a calcium wave, right? So we're making these little flashing beaters now that we used genome engineering to target this in at a safe harbor locus. So it's kind of fun just to see that, like for a screensaver, but we actually did this so that we could follow our cells after we transplanted them, because we wanted to know, are you live, are you beating, and what's the relationship of your beating to that of the native heart. So that's what you're going to see here. This is a black and white movie of the same sort of idea. We're looking at about the bottom 25% of a macaque monkey's heart. So so this is the left ventricle. This would be the area fed by the artery that we occluded. So this is the location of myocardial infarction. This is a still. I'll start the movie in a minute. But what we saw, these white streaky areas turned out to be big chunks of cardiac graft where the fluorescence is shining through the wall, and we can capture it with a camera. And it's kind of bright in here, but I think if you look up here, you can get the idea that there's a region of flashing. And then if you look over here, maybe you can see there's flashing in here and down here and over here and that sort of thing. And this whole zone actually is lighting up with cyclic fluorescence, and the cyclic fluorescence is all in phase. So what does that tell you? It's alive, it's beating, and the graft is at least synced up with itself. So that was pretty cool. When we went offline to do analysis on that, we saw what our eyes told us. Here are 3 different graft regions. It's just arbitrary fluorescence units for the flashing. And each of these 3 regions, you can tell they're all synced up. The part that was particularly cool is when we overlaid the electrocardiogram, you can see the QRS complex of each beat, of each heartbeat, is what is triggering this. So in this context, the monkey's pacemakers are calling the shots, and the human cells are responding in perfect synchrony. So this was a major breakthrough. There was much high-fiving and a few cocktails were celebrated, that sort of thing, because it was our first evidence that we had really the functional evidence of regeneration. At least we had electrical integration. So what about mechanical function of the heart? This may be the most important single slide that I show you. So the vertical axis here is that index of ventricular function that I mentioned, left ventricular ejection fraction. It's a measure of systolic function, of squeeze. And this, again, is how much blood is squeezed out of the left ventricle with each beat of the heart. And you can then, before myocardial infarction, monkeys are like healthy people. It's sort of in the 60 to 75% range. Blue is control. They got a saline injection. And red is the animals that received our human cardiomyocytes. And by the way, these animals were all heavily chemically immunosuppressed because, of course, their immune systems would reject these xenografts otherwise. And you can see the infarct, these big infarcts, we lost a lot of function, more than 20 ejection fraction points. Ejection fractions averaged about 40% or so. And then 4 weeks afterwards, there was minimal improvement in the animals that received a saline injection, whereas the animals that received the human cardiomyocytes, each and every one of them showed a fairly significant improvement in their ejection fraction. Another way to look at this is to use each animal as its own control. So you subtract the 4-week value, you subtract the baseline value from the 4-week value and say, what's the change in ejection fraction? Minimal change under 3% in the control. That's probably within error of measurement for an endpoint like this. And we had about 11% change in the animals that received our human cardiomyocytes. So you subtract the two, and arithmetically you get about an 8-point difference, 8 ejection fraction points. Now you could say, 8 points, is that a big deal? Is that a little deal? It's 8 points, right? Well, if you compare it to what's out there currently, standard of care for myocardial infarction, it's better than reperfusion therapy. It's better than beta blocker therapy, and it's better than angiotensin-converting enzyme inhibitors. And it's better than the 3 of those all put together, which is our current standard of care. So I would submit if we could do this in people, that'd be pretty good. But we wondered, well, we stopped most of these studies just 4 weeks out because they're logistically quite challenging. But maybe they weren't done getting better. And so these are the same data set. I just crunched them down a little bit, but that you saw previously. But we took 3 of these animals, 2 treated animals and 1 control animal, and we extended this out to 3 months afterwards just to see, A, is the benefit stable, and B, might it even get better? And so what you see here is the first part of the data set from those 3 animals, and you can see they're embedded within the data that you see here, and they behave according to the means of their groups and that sort of thing. But when we extended it out to 3 months, this is what we saw. The animal who was a control, no improvement whatsoever, and the 2 animals that were treated, essentially back to normal levels of ejection fraction. You wouldn't be able to tell that these animals had had an infarct by MRI scanning if you didn't know it in advance. So this is the thing that is getting me out of bed in the morning to come in and try to bring this to the clinic for patient therapy. Because I've been in heart research since 1984, and I've never seen anything that has an effect like this. This is what keeps me up at night, is the electrical instability. Macaque has a heart rate of about 120 beats a minute, a bit faster than a human. And this is telemetry data, so this is the normal sinus rhythm. Several days after engraftment, they often start to show spontaneous electrical instability, like this ventricular tachycardia. Here it's non-sustained, here it's sustained. And then we get this funky rhythm where it slows down. It's a wide complex rhythm that a cardiologist would call accelerated idioventricular rhythm. I think of it basically as slow V-tach. And the truth is, I think these 2 are basically the same arrhythmia, but just different rates. So that's what we wanted to try to figure out the mechanism of. So we went to mapping this. We did this by using clinical grade tools, which are kind of tricky to get into a monkey heart. A monkey heart's about the size of a plum. And so it's kind of like trying to get a big car into downtown Seattle or something like that. It wasn't exactly made for this, but we got a reasonable resolution map. Just to orient you here, first of all, anatomically, these three balls up here represent the cusps of the aortic valve. So this is the base of the heart in the upper left, and the lower right is the apex of the heart. And then the color gradient is, red is the first electrical activation, and the blue to purple is the last electrical activation. Now the heart should normally activate at the tip, at the apex. The electrical activity breaks out there, and then it spreads up towards the base, and of course that's so the heart can contract from apex to base because all the great vessels are at the base, and that's how normal physiology is. And from mice through whales, as far as I know, this is how the heart works. This map did not show, and this was an animal that came in spontaneous ventricular tachycardia. So we mapped this arrhythmia, and I think if the movie plays, you will see how it breaks out. It originates up here in the sort of midway from apex to base in the septum, probably. And that was funky. And it's doing this at like 220 times a minute, just going bing, 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 bing like this. So there are two general categories of things that cause arrhythmias. One are areas of disorders of impulse conduction, where you get a region of slow conduction, and by the time it breaks through, the other part of the heart is electrically excitable again, and you can get what's called a circus movement, where it goes round and around and around, and that's called re-entry, kind of like when people do the wave in the stadium, and it just goes around and around. And that's where my money was. I thought this was slow conduction within our Grafts zone causing re-entry. The other kind of arrhythmia is one of impulse generation. You can think of it like a pacemaker being in the tissue and firing, trying to take over the heart. And so we put these animals through the paces, literally, and did a number of interventions. And when we mapped it like you saw here, it was very focal. So it was either a very small rotor or there was no rotor at all. We did pacing where we could make the heart go faster. We could take 220 beats a minute and take it up to 300 beats a minute. That's not victory. But usually if it's re-entry, when you stop that, the normal rhythm takes over. And so that didn't happen. And then we shocked it with the DC cardioversion. And typically with re-entry tachycardia, that resets and sinus rhythm resumes. And the heart paused for about a second, and it just went right back to pinging away. So from these kinds of studies, our conclusion is that this is a disorder of impulse generation, not a disorder of reentry. And I guess that's why it's good to do experiments, because I was totally wrong in terms of what I thought was, my hunch was not particularly useful. So these guys are behaving like little pacemakers. So now I've had to learn all this electrophysiology, and I'm not going to get into that today, but this is what I think about every morning now when I get up. It's like, what the heck, how are we going to get around this? And I don't have the answer, so stay tuned for a year and see what happens. Let's look structurally what's going on. This, unfortunately, it's a little dim here, but this is a low magnification fluorescence micrograph of the monkey heart that's from a control animal that was infarcted. The red is monkey heart muscle, and the blue is scar tissue. And there is no human in this case, of course, because so what you see is this is the non-ischemic region, and then this is the region that's ischemic. It's a scar pretty much all the way through the wall, and this little bit of muscle that you see is residual muscle that survived. And the wall, you'll notice, has gotten quite thin. It's about a third the thickness of the healthy part over here. Let's take a look at the histology of an animal that received our cardiomyocytes. This is a low magnification scan of it. We have we have a beautiful human-specific antibody that recognizes a contractile protein isoform called troponin I. Chunks of new human heart muscle in the infarct and in the peri-infarct region. Note the scale bar. So this is like a centimeter or more of new muscle that we've been able to grow that's shown in this particular plane of section. So we have really successfully remuscularized a good chunk of the heart wall. So what are these cells if we look at higher magnification? There's at least 3 different kinds of cardiac muscle cells. There's atrial cells, ventricular cells, and pacemaker cells. And we would desire to repair the ventricle with ventricular cells, of course. The antibodies that we're going to use here, the red is ventricular, so this is a myosin light chain 2V. V stands for the ventricular isoform. 2A is the promiscuous isoform. It can't tell any of the three apart, and in fact, the immature ventricular cells would be green, and then when they mature, they would become red. Oh, I think this is going to help. Yeah, thank you. So 1 month after transplantation, this is the host cardiac muscle down here. The graft is up here. It's mostly red with a little green. That means it's mostly ventricular with a little maybe, right? 3 months afterwards, it's almost entirely red. We can find the rare green straggler. So if we quantify that, each one of these bars is one primate heart. You can see that it's 88% or so ventricular at 1 month, and it's 99% by 3 months. I think it's probably all pretty much ventricular, but it just has a time-dependent maturation where they have to go through this myofibrillar isoform switch. So that was good news. It seems like we have the right kind of cells. They mature over time. And I know that you're not all geeky cardiac aficionados like I am, and so I'll try not to make this boring. But you can see this is a double label where green is immature, it's the skeletal troponin isoform, and red is the mature, the human troponin isoform. At a month after transplantation, these you can hardly even tell this is muscle. They're kind of scraggly looking little cells, and it's mostly green. But 3 months, you can start to see that this is really looking like proper muscle. And it's not so green, and it's much more red. So it means that the cells are flipping in their isoform switch, and so they're expressing the mature cardiac troponin isoform. So that's good, and maybe this change from being scraggly little guys to being hypertrophic guys like you're seeing at 3 months is why function continues to improve, because the muscle cells are getting stronger. This is a yet higher magnification looking for a structure called T-tubules, which you may know are these invaginations of the sarcolemma that bring the depolarization into the depths of the cells. That's another good maturation marker, and we use this green stain for a protein called caveolin 3 to mark T-tubules, and we have beautiful T-tubules at this point. So it looks like they're almost adult in their phenotype at this point. The last bit of data that I'll show you is we were just pleased to see this, is we can get grafts oftentimes that are big enough to see by magnetic resonance imaging scanning. I'll show you the scans first. And so this is using a technique called delayed gadolinium enhancement. So gadolinium is a marker that gives you a bright signal. It marks the extracellular space. And so we're looking at the same animal and the same level of its heart at baseline after the infarct, 1 month after cells, and 3 months after cells. You see a dark region here, that's healthy muscle. You see a bright region here, this is the anterior wall and septum, and that's the region of the infarct where the cells have died off, and it's mostly extracellular space, so that's bright. What you see at 1 month after cell therapy is these dark blobs that are emerging here. So something is excluding gadolinium from the extracellular space. There's no extracellular space there, and this persists at 3 months. Our MRI person brought this to me, and she said, I think we're seeing grafts. And I'm like, yeah, right. And I said, look, we've got the histology on these. We should be able to, if we look at the region that corresponds to this, we better see a big honking graft if we're going to make this claim. And this is the histology that corresponds to this. This was a big chunk of human myocardium at this level that we're seeing. And again, you notice the scale here is 5 millimeters. This is 1 centimeter here. So looks like we're probably able to see it, at least in some animals, that we're making enough new myocardium in the scar that we can actually see it by scanning. And of course, you can see tumors that are this big by magnetic resonance imaging, so it makes some sense. So, what have I told you? Basically, you can make human cardiomyocytes in bioreactors in essentially unlimited amounts from pluripotent stem cells. It's really just money and people. And you can transplant them and remuscularize the heart of a pretty large animal, in this case, macaque monkeys. We never see teratomas. We pay a lot of attention to the care of our cells and their differentiation, but we've done thousands of small animals, and we don't see tumors growing from this. It clearly improves function of the heart at 4 weeks after transplantation. We've got good statistics to back that up. And we see further improvement in a subset of animals that went out to 3 months. I think that's real, but I can't give you a p-value for it. They're mostly ventricular cells, and they make the heart a little twitchy. And that's the biggest challenge to taking this forward right now. But at least we have insights into the mechanism which look like ectopic pacemaking from the grafts generating their own rhythm. So I don't know of any reason why human cardiomyocytes would be better for monkeys than they would be for people. And so I think probably if we could do this in a human being, we would get a good response functionally in a human being as well. And we may get the rhythm problem as well, too. So it's the double-edged sword at the moment. But I think certainly these cells have the potential to improve human cardiac function. So from the standpoint of pushing this into the clinic, we've developed a potential therapeutic that's got a different mechanism of action from other kinds of cell therapies that have been done so far. It engrafts long-term. I think this would go for the life of the animal if you can keep the immune system at bay, and it beats synchronously. So this makes it different from all the other kinds of cardiac cell therapies that people have developed. And to take this forward, what's key? We need to come up with a less invasive therapy. This way to deliver. This was done surgically. We've got this working with catheters in pigs now, though, so we can do that. So there's a check mark there. We've got to get around the immune system. If people are curious, we can talk about that. But nobody wants to have this level of immunosuppression after they've had a myocardial infarction. But I think there's a good path forward on that. And we need to be able to control the graft-associated arrhythmias. So with that, I would like to just say thank you for your attention. Thank you for inviting me to speak here. What an interesting society this is. As I mentioned, this whole thing's been a big team effort. We wouldn't get here without many other people from our Primate Center, our Cardiovascular Institute, and that sort of thing. Thank you for paying your taxes, because we're all on the dole from NIH as well. And with that, I'll stop and take any questions if there's time. Thanks very much.